Chapter 12 Biotechnology Regulations. FDA Inspector at Work. The FDA is one of the government agencies responsible for evaluating the safety of biotech products. After completing this chapter, you should be able to describe the Animal and Plant Health Inspection Service, APHIS, and the U.S. Department of Agriculture's permitting process, including the precautions that must be taken to prevent accidental release of bioengineered plants into the environment. List the 6 criteria that must be met before a plant can be eligible for notification under APHIS guidelines. Describe the role of the Environmental Protection Agency, EPA, in regulating biotechnology products. Describe the role of the U.S. Food and Drug Administration, FDA, in regulating food and food additives produced using biotechnology. Describe the FDA's role in regulating pharmaceutical products, including phase testing. Cite examples of the regulatory agency's ability to respond expeditiously to emerging situations. Describe the function of patents in science and explain how patents encourage discovery. Describe the circumstances in which DNA sequences are patentable. The use of a mislabeled drug or consumption of a contaminated food can cause death. Medicines and food are regulated by the government. These regulations have profound effects on the biotechnology industry, especially in the United States. It can take years to test a new drug to confirm its safety and effectiveness. In the meantime, lives may be lost that the new drug might have saved. There is a cost if food must be discarded, just as there is a cost if agricultural productivity is diminished because potentially toxic herbicides are not used. Clearly, there is a conflict between ensuring safety and reducing costs. New and innovative biotechnology products in medicine and agriculture promise benefits but also pose risks. When former President Bill Clinton and UK Prime Minister Tony Blair pledged to limit gene sequence patents by, among other things, sharing information gathered by researchers working on the Human Genome Project, panic spread in the biotech industry. Many applaud a restriction on the patenting of gene sequences because they argue that such patenting is morally unacceptable. At the heart of this debate is what it means for something to be patentable. Your opinion about that may change once you have read about protections and patents in this chapter. Forecasting the future. Biotechnology pioneer Craig Venter, a primary contributor to the original human genome sequencing effort, has played a key role in establishing bioethical regulations for synthetic genomes in the industry. When the first gene transfer experiments occurred in the 1970s, society's fear that a genetic monster would be created resulted in a voluntary moratorium of experiments until safeguards were established, safeguards that Venter requested in the 1990s before his team transplanted a synthetic genome into a bacterium. Venter and other biotechnologists knew that without such safeguards, their experiments might also be stalled owing to society's fears of genetic engineering. Because of Venter's initial efforts to establish safeguards and regulations, the results of his original experiments are being used as the basis for future work crucial to biotechnology, a designed alga that can make new hydrocarbons from CO2 and be used in refineries, the speeding up of vaccine production, the cleanup of polluted water, and the production of new food chemicals, for example. These experiments will undoubtedly lead to even more new federal regulations and protections, but the groundwork for experimentation coupled with safety regulations was laid in the 1970s. 12.1 The regulatory framework. During the 19th century in America, peddlers freely promoted their patented medicines, but often their main concern was to skip town with their profits before local authorities could catch on to the fact that most of these so-called medicines were scams. At the same time, many large companies were poisoning the environment, often without any real understanding of the long-term consequences of their actions. Eventually the government could no longer ignore threats to public safety from either small or large businesses. As a result, it established protective agencies. The new federal agencies were charged with overseeing the safety of food and medicine and protecting the environment from polluters, and these agencies regulate those same industries today. By the time biotechnology emerged as a field of research, a regulatory system to oversee the industry was already in place. The 1974 Asilomar Conference was a voluntary meeting to establish guidelines initiated by researchers themselves when some of those involved in gene transfer saw the potential for spread of disease genes if safeguards were not put in place. The National Institutes of Health, NIH, was the first federal agency to assume regulatory responsibility over biotechnology. Among other things, the NIH published research guidelines for recombinant DNA techniques. Recall that the Recombinant DNA Advisory Committee of the NIH sets the guidelines for working with recombinant DNA and recombinant organisms in the laboratory, Chapter 3. The NIH continued to monitor and review all DNA research until 1984. At that point, it became clear that a different set of regulations was needed. The government published the Coordinated Framework for Regulation of Biotechnology in 1986, which made biotechnology research a joint responsibility of the NIH, the USDA, and the EPA. It describes the federal system for evaluating products developed using modern biotechnology. This groundbreaking report proposed that biotechnology products should be regulated much as traditional products were. For example, bioengineered plants would be monitored by the same agencies that regulated other plants. Bioengineered drugs would fall under the same rules that applied to all other drugs. In essence, this report proposed that biotechnology products would not pose regulatory or scientific issues that were substantially different from those posed by traditional products. This concept is central to understanding the regulation of biotechnology in the United States. The framework is based on health and safety laws developed to address specific product classes. 3 agencies are responsible for regulating most of the products that are the result of biotechnology: the U.S. Department of Agriculture, USDA, the Environmental Protection Agency, EPA, and the U.S. Food and Drug Administration, FDA. The USDA makes sure that a given organism is safe to grow. The Table 12.1 Primary federal regulatory agencies in the United States. Source: www.fda.gov. Regulatory Oversight of Biotechnology Products Agency. Product Regulated. U.S. Department of Agriculture. Plants, plant pests, including microorganisms, animal vaccines. Environmental Protection Agency. Microbial/plant pesticides, other toxic substances, microorganisms, animals producing toxic substances. U.S. Food and Drug Administration. Food, animal feeds, food additives, human and animal drugs, human vaccines, medical devices, transgenic animals, cosmetics. Major laws that empower federal agencies to regulate biotechnology. Law. Agency. The Plant Protection Act. USDA. The Meat Inspection Act. USDA. The Poultry Products Inspection Act. USDA. The Eggs Products Inspection Act. USDA. The Virus Serum Toxin Act. USDA. The Federal Insecticide, Fungicide, and Rodenticide Act. EPA. The Toxic Substances Control Act. EPA. The Food, Drug, and Cosmetics Act. FDA, EPA. The Public Health Service Act. FDA. The Dietary Supplement Health and Education Act. FDA. The National Environmental Protection Act. USDA, EPA, FDA. EPA makes certain that it is safe for the environment, and the FDA determines whether it is safe to consume. See Table 12.1. These 3 agencies cooperate to achieve their goals, and all are often involved in the oversight and regulation of a single biotechnology product. Even though bureaucracies are often depicted as slow to change and act, it is important to understand that the regulatory process is constantly being refined to ensure that products, both bioengineered and traditional, are safe. 12.2 U.S. Department of Agriculture. Created in 1862, the USDA has many functions related to the advancement and regulation of agriculture, including regulating plant pests, plants, and veterinary biologics. A biologic is broadly defined as any medical preparation made from living organisms or their products. Insulin and vaccines are examples of biologics. Animal and Plant Health Inspection Service. The Animal and Plant Health Inspection Service, APHIS, is the branch of the USDA responsible for protecting U.S. agriculture from pests and diseases. Because genetically engineered plants are potentially invasive, they are treated as plant pests and regulated by the agency under the requirements of the Federal Plant Pest Act. APHIS provides permits for developing and field testing genetically engineered plants. If an experimental organism poses a potential threat to pre-existing agriculture, the agency makes certain that safeguards are in place. APHIS regulatory process. After a new plant has been engineered, APHIS requires several years of field trials to investigate issues such as disease resistance, drought tolerance, and reproductive rates. APHIS will not approve a field trial application unless precautions are taken to prevent accidental cross-pollination. For example, trial planting sites for transgenic corn must be at least 1 mile away from fields where seed corn is grown and harvested. A 25-foot perimeter of fallow, unplanted, tilled land must surround the entire trial site. In some cases, the tassels at the ends of the ears of corn must be bagged to confine the pollen, as seen in Figure 12.1, and the tassels themselves must later be removed. Plants must not escape the trial site, which means that plants that naturally reseed must be destroyed and that no plants can be removed unless they are to be destroyed. The entire trial site is usually kept fallow for the season following the trial harvest. APHIS employees inspect the sites before, during, and after trials to make sure that all requirements are followed. An example of this protection can be illustrated by the fact that in 2002, 2 growers in Iowa and Nebraska were required to destroy all the seed corn produced from fields where they had previously grown transgenic soybeans for protein, human antibody, extraction, but did not remove a few volunteer plants that could have contaminated the subsequent seed corn crop, as required by the USDA. They were also fined $250,000 each for this infraction. The case illustrates that the regulators are watching and the regulations are being enforced. The APHIS approval process. Of course, the ultimate objective is to harvest a marketable product. The first step in that process is to petition APHIS for deregulated status. Genetically engineered plants with this status are monitored in the same way as traditional plants. APHIS reviews field test reports, scientific literature, and any other pertinent records before it determines whether the plant is as safe to grow as a traditional variety. APHIS considers 3 broad areas while evaluating the petition for deregulation. Plant pest consequences. APHIS examines the biology of the plant to evaluate the possible threat to other plants. This is known as the plant pest risks. The agency investigates the possibility that the new genetic material might lead to a new plant disease. It also considers whether crossbreeding with native plants could create new superweeds. Risks to other organisms. The agency investigates the risk to wildlife and desirable insects that might feed on the crop or be exposed to the pollen. Weed consequences. Finally, the agency considers the possibility that the new plant will be unwelcome and invasive, in other words, a weed. The plant's reproductive strategies are an especially important consideration. A plant that reseeds itself easily, has a great mechanism for spreading its seeds, and is resistant to cold and drought can present a real problem in the future. For instance, if the dandelion were presented as a new biotechnology plant, it would be rejected on the basis of its amazing success in propagation. Figure 12.1 APHIS regulations in action. Corn growing in a field with its tassels bagged to prevent pollination of unintended plants in nearby fields in compliance with regulations from the Animal and Plant Health Inspection Service of the USDA. The APHIS notification process. An alternative system called notification is in place to fast-track some new agricultural products. 6 criteria must be met before notification becomes an option. The new agricultural product must be one of only a limited number of eligible plant species. These species including corn, cotton, potatoes, soybeans, tobacco, and tomatoes have all been thoroughly studied in past trials, and most of their characteristics are well understood. The new genetic material must be confined to the nucleus of the new plant. It cannot be floating in the cell on plasmids or viral vectors. Nuclear genes tend to remain with the new engineered plant. See Chapter 3 for a refresher on vectors used in the genetic engineering of plants. The function of the genes being introduced must be known, and no plant disease can be caused by the protein being expressed. If the new agricultural product will be used for food, the new genes cannot cause the production of a toxin, an infectious disease, or any substance used medically. If the gene is derived from a plant virus, it cannot have the potential to create a new virus. The new genetic material must not be derived from animal or human viruses, to prevent spread as a new human disease. If a bioengineered plant meets all 6 of these criteria, the agency will approve the field trial within 30 days. Under notification, the plant developer is still required to meet the standards demanded under the permitting process. The same precautions must be taken to prevent accidental spread of the new plant, and detailed records of the trial must be maintained. After the safety of the new plant has been determined, a grower can cultivate, test, or use the plant for crossbreeding purposes without monitoring or approval by APHIS. The new biotechnology product can then be brought to market. But these requirements may not be all that is needed, and the EPA regulates all new products introduced into the environment. 12.3 The Environmental Protection Agency. Established in 1970, the EPA has responsibilities ranging from protecting endangered species to establishing emission standards for cars. Another major duty is regulating pesticides and herbicides. As a natural extension of that responsibility, the EPA regulates any plant that is genetically engineered to express proteins that provide pest control, Chapter 6. The EPA also supervises the use of herbicide-tolerant plants, Figure 12.2. In this case, the EPA is interested not only in the plant but also in the herbicides that will be used in the fields where the herbicide-tolerant plants are grown. Of particular importance is the pesticide residue remaining on the crop. The EPA monitors these levels to make certain that the crop itself is safe for human consumption. Both USDA, APHIS, and EPA approvals are needed. Figure 12.2 Ice-sprayers protected by the EPA. Moon suits, like the ones illustrated here, are used to protect the sprayers against unknown biohazards while they apply ice-bacteria to strawberry plants to reduce freezing. Full description of ice-bacteria is in Chapter 5. Experimental use permits. Plant developers who intend to create a plant that expresses pesticidal proteins must first contact the EPA. Field experiments involving 10 acres or more of land or 1 acre or more of water cannot be conducted without an experimental use permit, EUP, issued by the agency. EUPs require careful record keeping because the plant cannot be registered for use as a pesticide without clear evidence that there will be no unreasonable adverse effects as defined by U.S. pesticide law. In other words, the plants themselves must meet the same standards as chemical pesticides used in the fields. Deregulation and commercialization. The next phase of the process is commercialization, during which the product is approved for market. However, before anything is available for sale, the EPA spends about a year reviewing the data collected during experiments. This review concentrates on 4 general areas of concern. First, the EPA considers the source of the gene, how it is expressed, and the nature of the pesticide protein produced. Second, the health effects of the bioengineered plant are studied in depth. Experimenters use animal models to measure the impacts of the protein on living organisms, including how easily the protein is digested and whether it can trigger allergies. Third, because the EPA is responsible for protecting the environment at large, the agency also must investigate the environmental fate of the pesticide protein. One concern is the rate of degradation of the pesticide and whether it lingers in the soil or water. Another issue is the possibility that the pesticide protein might escape through cross-pollination with weed plants to create a superweed. Fourth, the EPA is interested in the effects on non-target species, which can include helpful insects such as honeybees and ladybugs as well as other animals including fish, birds, and rodents. Chapter 6. Like the USDA, the EPA can grant deregulated status to any plant that meets its requirements. Any engineered plant with deregulated status is registered with the EPA and can be sold or distributed like any other plant. But even in these cases, the mission of the EPA is not complete. The agency must be prepared to respond to new information, and it has the power to amend or revoke existing regulations whenever required. Chapter 6. The bureaucratic regulatory framework in the United States is responsive to emerging public concerns and proceeds with caution. Perhaps most importantly, however, the regulatory bodies depend on good scientific evidence to come to their conclusions. The public is similarly protected by regulations relating to the release of foods and drugs. 12.4 The U.S. Food and Drug Administration. The FDA is charged with making certain that the foods we eat and the medicines we use are safe and effective. Because both food and drug products are hot sectors of biotechnology development, the FDA is busy monitoring new developments in the industry. They make sure that biotechnology products are as safe as their conventional counterparts. Food and food additives. When a new food product or additive is being developed, the FDA serves as a consultant to biotechnology developers, companies, university researchers, etc., and advises them on testing practices. Studies focus on whether the new product has any unexpected, undesirable effects. The expressed protein is evaluated to see if it is substantially the same as naturally occurring protein in food. Because many common foods including milk, shellfish, peanuts, and eggs are known to contain allergy triggers, any protein derived from such sources must be considered a potential allergen and labeled accordingly. Companies are not required to consult with the FDA in developing biotechnology food products that are already on the market. However, most companies in the United States have taken advantage of the FDA's expertise before bringing a food product to market, because if a food product or additive poses no foreseeable threat, the FDA can grant generally recognized as safe, GRAS, status, which may help to give the product a positive reception when it reaches the market. An example of a product with GRAS status is genetically engineered chymosin, which is used in 80% of the cheese manufactured in the United States. Chapter 4. Sometimes a product that has been deemed safe by the FDA will still have a tough time in the marketplace despite this. An example is the use of bovine somatotropin, BST, which is injected into dairy cattle to increase their production of milk. Although BST occurs naturally in cows, consumers have concerns that it will show up in milk sold in stores. The debate continues as to whether there are negative effects on cattle or humans who consume the milk so produced. No hard data suggesting that BST presents any danger have been uncovered, even after an 8-year study by the FDA. However, if BST proves to be unsafe, the FDA has the responsibility and power to prevent its use. The FDA drug approval process. The FDA is also responsible for regulating new pharmaceutical products. Biotechnology drugs must meet the same exacting standards as any other new drug. Imagine for a moment that a new biotechnological treatment for cystic fibrosis has shown promising results during animal trials. The first step to getting the treatment to patients would be to contact the FDA and file an investigational new drug, IND, application with the Center for Drug Evaluation and Research. Before approving the substance for testing in human patients, the FDA would consider the results of previous experiments, the nature of the substance itself, and the plans for additional testing. To provide effective regulatory review of biological products, the Center for Drug Evaluation and Research conducts active mission-related research programs. This research greatly expands knowledge of fundamental biological processes and provides a strong scientific base for regulatory review. Phase testing of drugs. If the FDA is satisfied that a new drug warrants further investigation, does not present obvious risks, and will be tested using scientifically sound methods, it attains IND status. The next step is phase testing, which is more stringent than the testing that foods undergo. First, during phase I, safety, between 20 and 80 healthy volunteers take the medicine to see if there are any unexpected side effects and to establish the dosage levels. Phase II, efficacy, begins the testing of the new treatment on 100 to 300 patients who actually have the illness the drug is designed to treat. If no detrimental side effects are noted and the drug seems to have some positive effect, it is ready to go to phase III testing to assess its benefit compared with other drugs. This phase involves between 1,000 and 3,000 patients in double-blind, where neither the patient nor the clinician knows whether placebo or drug is being administered, so as to remove potential bias from affecting the results. Tests and lasts for 3.5 years. Generally, a drug can be marketed only after its benefits and long-term safety have been established in phase III studies. Most drugs never make it this far. Only 20% of drugs that go through phase I testing make it all the way to phase III and on to FDA approval as an NDA, New Drug Authorization. No therapeutic drug product can be marketed in the United States until an NDA has been issued by the FDA. Even after a new drug enters the market, it continues to be monitored, phase IV, by the FDA indefinitely. The number of new drugs that the FDA has approved has fallen by half since 1996, with only 20 approved in 2005. Phase III trials consume 70% of clinical development costs. Yet 40% of compounds now fail at this final hurdle, largely owing to the inability to demonstrate superiority over placebo. See Figure 12.3. A biotech company will file for a biological license agreement, BLAT, similar to an NDA, if they are seeking approval of a biologically derived product such as a viral therapy, blood compound, vaccine, or protein derived from animals. The FDA reviews information that goes on a drug's professional labeling and, as part of the approval process, the FDA inspects the facilities where the drug will be manufactured. Exceptions to phase testing. An important exception to this testing procedure must be noted. The FDA does permit the approval of drugs and vaccines intended to counter biological, chemical, and nuclear terrorism without first proving their worth in phase II and III trials. This is also true for orphan drugs, as designated by the FDA Office of Orphan Products Development, OOPD. These are drugs with small numbers of beneficiaries but with great benefit, such as human botulism immune globulin, BIG, an antibody for botulism poisoning. Because it would be unethical to expose human beings deliberately to smallpox or nerve gas to test the value of such treatments, there are no pools of patients on which these treatments could be tested. The OOPD administers the major provisions of the Orphan Drug Act, ODA, which provide incentives for sponsors to develop products for rare diseases. The ODA has been very successful. More than 200 drugs and biological products for rare diseases have been brought to market since 1983. In contrast, the decade prior to 1983 saw fewer than 10 such products come to market. New drugs of this kind will still undergo phase I testing because that part of the process requires exposure to the drug only, not to the dangerous substance it is meant to counteract. Therefore, the FDA can determine that the drug itself will not cause unwanted side effects. For example, the FDA expedited approval of the drug ciprofloxacin, an antibiotic used to treat anthrax. The development of good laboratory, GLP, clinical, GCP, and manufacturing, GMP, practices. In 1975, FDA inspection of a few pharmaceutical testing laboratories revealed, among other problems, poorly conceived, carelessly executed experiments, inaccurate record keeping, and poorly maintained animal facilities. These deficiencies led the FDA to institute the Good Laboratory Practice, GLP, and Good Manufacturing Practice, GMP, regulations to Figure 12.3 The drug pipeline. Only 20% of the drugs that enter phase I will make it to phase 3, and 40% of these will fail due to the inability to show superiority over placebo. Govern animal studies of pharmaceutical products. GLPs require that testing laboratories and manufacturers follow written protocols, standard operation procedures, or SOPs, have adequate facilities, provide proper animal care, record data properly, and conduct valid toxicity tests. A similar set of standards, good clinical practices, GCPs, protect the rights and safety of human research participants and ensure the scientific quality of clinical experiments. Faster drug approval versus public safety. Biotechnology companies spend about $1.2 billion to bring a biological product to market, and it takes 8 to 15 years to receive FDA approval for marketing. Because of financial concerns, biotechnology companies would like to speed up this process. However, the FDA would prefer to keep the process at its present pace to guarantee adequate testing. A compromise may be needed to provide products that the public needs, such as gene therapies to treat fatal diseases, while maintaining safety that is acceptable to regulatory agencies like the FDA. For instance, there are currently 5 suppliers of human growth hormone. Each company was required to get individual approval from the FDA, each doing its own phase testing. The U.S. Pharmacopeia Convention, USP, has proposed an alternative method whereby the 5 companies could share early portions of the testing. This change would mean that each of these companies could accelerate the testing and proceed to the human safety trials a little earlier. From the point of view of the pharmaceutical companies, this change in methodology would address the backlog of products being submitted for approval by using standardized methods without compromising safety. Whether this change will be approved can only be determined by time. Adaptive phase testing for faster results. In an attempt to get quicker results with smaller numbers of individuals in clinical drug trials, the FDA is encouraging drug companies to design clinical trials with flexible enrollment and dosing. Not everyone agrees that this is a good idea. In a typical clinical trial, drug dosage and the number of patients in the control and experimental groups are predetermined and unchangeable. In contrast, adaptive trials allow changes in drug dosage, patient pool sizes, and other changes in response to incoming data. In other words, the parameters are changed on the fly as new results are received. Considering that phase 2 and 3 trials can easily drag on for 5 or more years, everyone is supportive of changes that will possibly speed results. Bayer Healthcare used an adaptive approach in its phase 2 trials for a new cancer drug. They did not know ahead of time which cancer would respond best to the new drug, so they enrolled a number of patients with different advanced cancers. Within a short time they knew that the drug responded best to kidney cancer, so they adapted their trials to enroll and test only patients suffering from advanced kidney cancer. The major concern is that the committee to decide on adaptive responses should be completely objective. It is quite possible that drugs in trials will fail faster and cost companies more, and results could be manipulated with the right adaptations. The FDA is releasing guidelines for evaluating multiple possible outcomes and methods to enrich trials with patients who are most likely to benefit. Will adaptive design provide faster results? Will the cost of drugs go up or down? Who will benefit the most, the patient or the drug company? FDA regulations to protect laboratory workers. Laboratory workers who handle gene vectors, recombinant DNA, and biological organisms containing recombinant DNA, bacteria, and fungi risk infections as a result of their work. Biotechnology companies are aware of the potential liabilities and financial implications related to employee infection and have made implementation of the appropriate biosafety practices a high priority. There are 3 levels of protection against biological hazards. A Class 1 cabinet protects the operator from airborne material generated at the work surface, where the air flows over the work surface and is filtered before being vented back into the room. A Class IIB cabinet draws high-efficiency particulate, HEPA, filtered air across the work surface, refilters the air before venting into the room, and is suitable for bacterial and tissue cultures. A Class 3 cabinet isolates the material completely in a gas-tight area, which is required only in working with the most hazardous agents. Figure 12.4 on the next page. 12.5 Legislation and Regulation. The ongoing role of government. The regulation of biotechnology is a matter of politics as well as science. Regulatory agencies are government organizations, and they exist to enforce laws. Ideally, those laws rest on good scientific practice, but they also reflect philosophical values. It is not uncommon. Figure 12.4 Lab technician uses a micropipette under biosafety level 1 conditions. For value systems to clash, and when they do, a long process of negotiation and compromise may be needed to reach a consensus. Labeling biotechnology products. A controversial issue is the labeling of foods that contain genetically modified organisms, GMOs. From the perspective of the FDA and the current White House administration, food labels must include information about the ingredients and any claims made or suggested by the manufacturer. The FDA also requires special labeling of foods that present known safety, usage, or allergen issues. If a biotechnology food product included a protein that was not usually found in the food and was a known allergen, the FDA would require special labeling to notify allergic consumers of the risk. Many chocolate candies include a warning that they may contain traces of peanuts. However, because biotechnology foods are not inherently more or less dangerous than their traditional counterparts, the FDA does not require labeling to indicate the method of production. If the product is to be sold in Europe, however, the European Union does require labeling when genetic modification has occurred. Some consumer groups are pursuing a change in labeling laws that would require information about the use of genetic engineering in food production. They argue that this disclosure is essential to their right to know and to make informed choices based on their own values and beliefs. An interesting example of this real-world debate focuses on the use of BST to increase milk production, as discussed earlier. Over the past decade, a series of comprehensive studies have indicated that milk produced using BST presents no significant health risks. For this reason, the FDA continues to hold the position that no label is required. Furthermore, when dairy producers began labeling their products as BST-free, the FDA intervened, stating that such labels are misleading because milk production in dairy cows depends on naturally occurring BST. This has spurred sales of milk labeled from cows not treated with BST. Some people fear that the introduction of proteins into foods will stimulate allergies or otherwise add antibiotic-resistant substances to our foods. See Chapter 7. Safeguarding against potential hazards is what the regulatory agencies have been doing for decades. The future is likely to present many more debates as society adjusts to the role of biotechnology in our world. For this reason, one of the primary responsibilities of all regulatory agencies is to provide an avenue for public comment. This aspect of regulation is an important element of democracy in action and is shared by more than one agency, as you can see in Table 12.2. You can learn more about the role of public comment in shaping policy, or even participate yourself, when you visit the regulatory agency websites listed on the companion website. 12.6 Introduction to Patents. A patent gives an inventor or researcher exclusive rights to a product and prohibits others from making, using, or selling the product in these broad categories. Utility patents protect useful processes, machines and manufactured goods. Examples are computer hardware and medicines. Design patents protect against the unauthorized use of new and original designs for manufactured goods. Examples are the look of an athletic shoe or a bicycle helmet. Plant patents protect invented or discovered asexually reproduced plant varieties. To be granted a patent, a discovery must meet 3 basic requirements: it must be novel, it must be non-obvious, and it must have some utility. The concept of utility is discussed in more detail later in this chapter. Traditionally, patents have been awarded for inventions, but many natural products clearly meet all 3 requirements and fall into one of the categories described above. For example, a newly discovered DNA sequence can be novel, non-obvious, and potentially very useful, and the U.S. Patent and Trademark Office, USPTO, which oversees patents, generally does not hesitate to issue patents on sequences for proteins of known function. Table 12.2 Examples of shared responsibilities by federal regulatory agencies. New trait/organism. Regulatory review conducted by. Reviewed for. Source: www.fda.gov. Viral resistance in food crop. USDA. Safe to grow. EPA. Safe for the environment. FDA. Safe to eat. Herbicide tolerance in food crop. USDA. Safe to grow. EPA. New use of companion herbicide. FDA. Safe to eat. Herbicide tolerance in ornamental crop. USDA. Safe to grow. EPA. New use of companion herbicide. Modified oil content in food crop. USDA. Safe to grow. FDA. Safe to eat. Modified flower color in ornamental crop. USDA. Safe to grow. Modified soil bacteria that degrade pollutants. EPA. Safe for the environment. When patent laws were first conceived, no one could have imagined patenting DNA or proteins. It was expected that inventors would be engineering better mousetraps, not better mice. The USPTO issued the first patent on a bacterium with a unique gene sequence, genetically engineered organism, in 1980. Since that time, the office has granted more than 2,000 patents for plant, animal, and human genes. Patents have also been granted for transgenic animals and plants, monoclonal antibodies and hybridomas, isolated antigens and vaccine compositions, and methods for cloning or producing proteins. These patents have translated into big profits for biotechnology companies, but they have also stirred considerable controversy. Many people believe that genes, especially human genes, should not be treated like mousetraps. To their way of thinking, it should not be possible to patent any component of human life such as DNA and proteins. The process of patenting genes fully fits the letter and spirit of patent law. Still, many concerns and uncertainties remain. The challenge for the government is to find a way to protect the rights of individual researchers while promoting science as a whole. Obtaining a patent. In the United States, patents are enforced for up to 20 years from the earliest date of filing. That means other companies can market their own versions of the product after the original developers have had exclusive rights for 2 decades. This plan both encourages new discoveries and prevents long-term monopolies. You decide clinical trials for investigational drugs. If you were diagnosed with a serious life-threatening disease, would you want to be part of a clinical trial for a new drug? New FDA regulations announced in September 2010 improve your chances of benefit. The FDA has issued new rules to clarify the safety information that must be reported during clinical investigations of drugs and biologics. The agency now requires that certain safety information that previously was not required to be reported to FDA be handed over within 15 days of a safety issue turning up. Investigators will have to divulge findings from clinical or experimental studies that suggest a significant risk to study participants, serious suspected adverse reactions that occur at a rate higher than expected, as well as serious adverse events from studies that determine what percentage and at what rate a drug is absorbed by the bloodstream. They must also provide studies determining whether a generic drug has the same effectiveness as the brand name drug. These new rules are designed to protect the patient without slowing the rate of drug discovery. Are the risks of a new drug worth trying with new regulations in place? You decide. Source: FDA doles out new rules on clinical trial safety data reporting, Gen. September 29, 2010. To obtain a U.S. patent, a company must file an application that adequately describes the product. The company must also disclose what it considers to be the best use of the product at the time that the application is filed. USPTO experts with specialty technical backgrounds examine all patent applications. Patents are awarded on a first-come, first-served basis. If two companies file applications for the same discovery at around the same time, the two sides could be headed for a legal battle. In such cases, a little preparation goes a long way. The following guidelines protect a product that is heading for patenting. Record keeping. Every researcher should keep detailed notes within a bound notebook or in a secure electronic format as specified in FDA regulations. Each entry should be signed, dated, and witnessed by an individual who is not directly involved in the research. The notebook should contain all conceptual ideas and supportive data. This evidence may be used to support the company's patent or to invalidate rival patents. Preparation. Researchers should continue to monitor the activities of potential competitors and others in their field through trade literature, published patent applications, and issued patents. The USPTO website is a valuable source of information. You can also check commercial databases such as Derwent, Medline, and Biosis. Tools of the trade: a safe and efficient regulatory work environment. What can a group of well-trained scientists with excellent government funding, limited regulations, and a large supply of germ plasma produce? Chinese scientists have created the largest plant biotechnology production capacity outside of North America. They have shown that government support is essential if new products are to be developed quickly in developing countries. As a result, farmers in China are cultivating more acres of genetically modified plants than any other developing country, and they are doing this with the support and encouragement of their government. These crops are monitored and regulated by the Chinese government under regulations developed within the country. Although China has spent the past 50 years building the most successful agricultural research system in the developing world, employing more than 70,000 scientists, research in modern plant biotechnology did not begin until the mid-1980s. In response to rising pesticide use and the emergence of a pesticide-resistant bollworm population in the late 1980s, Chinese scientists began research on genetically modified cotton. In 1997, after devastating pest-related crop losses, the commercial use of genetically modified cotton was approved. The response by China's poor farmers to the introduction of Bt cotton eliminated any doubt that genetically modified crops can play a role in developing countries. By 2000, farmers planted Bt plant varieties on 20% of China's cotton acreage and reduced an average of 13 sprayings, and a savings of $762 per hectare per season. Although yields and the price of Bt cotton and non-Bt varieties were the same, the cost savings and reduction in labor enjoyed by Bt cotton users reduced the cost of producing a kilogram of cotton by 28%. A simplified approval process is not the only reason for the Chinese success. Chinese scientists have identified more than 50 plant species and more than 120 functional genes that scientists are using in plant genetic engineering. Of these genetic technologies, 353 were developed between 1996 and 2000. China's Office of Genetic Engineering and Safety Administration approved 251 cases of genetically modified plants for field trials, environmental releases, or commercialization. Of these, regulators approved 45 genetically modified plant applications for field trials, 65 for environmental release, and 31 for commercialization. Transgenic rice, resistant to 3 of China's major rice pests, stem borer, plant hopper, and bacterial leaf blight, have passed at least 2 years of environmental release trials. Unlike the rest of the world, in which most plant biotechnology research is financed privately, China's government funds almost all of its plant biotechnology research and planned to raise these research budgets by 400% before 2005, although they fell somewhat short of their goal. Some U.S. companies have found this favorable regulatory environment an enticement. Monsanto, based in St. Louis, Missouri, is pursuing a joint venture with the Hebei Provincial Seed Company in China to develop other plant varieties. China is vigorously pursuing opportunities for contract research, and it has become an exporter of biotechnological research methods and commodities. The sales of genes, markers, and other tools, in addition to genetically modified varieties, may cause other countries with more restrictive policies regarding genetic modification to look at this world leader. The value of patents in the biotechnology industry. For the biotechnology industry, strong patents mean strong businesses. Patents are the primary method by which a successful biotechnology company will be valued during all stages of its development. Suppose a team of scientists discovered a vaccine against a deadly virus. Without a patent, no pharmaceutical company would go forward with the daunting tasks of conducting costly clinical trials, obtaining FDA approval, and developing a comprehensive marketing plan. Without patent protection, a rival company could buy the drug off the shelf, duplicate it, and sell it at a discount without spending a dime on research. Patenting DNA sequences. Although the vast majority of DNA sequences may never have medical or agricultural value, some could play an important role in producing food or fighting disease. Naturally, organizations that identify those sequences want to enjoy the fruits of their efforts by obtaining protection under the patent laws. The genomic frontier has been mapped, and today's pioneers are eagerly and aggressively staking out what they regard as their territory. Under patent guidelines, applicants must assert a utility for the claimed invention that is specific, substantial, and credible. To claim a specific utility in the case of a DNA sequence, a researcher must know exactly what the DNA sequence does. It is not enough to say that it is a probe or a marker, see Chapter 3. The researcher must disclose what, precisely, it probes or marks, for instance, a probe for a specific human disease gene. A substantial utility defines a real-world use, for example, a cloned DNA fragment encoding a protein with a known function or use. To meet the credible utility requirement, the researcher must convince the patent office that the application is backed by sound science. Even if a gene sequence is proved to have utility, it can still be denied a patent if it is too similar to an existing patented sequence. Remember that the function of a gene can be dramatically altered by switching a single nucleotide, Chapter 3. For this reason, patents on gene sequences are usually very specific. Put another way, a segment of DNA usually must match a patented sequence exactly in order to violate or infringe on that patent. If a company claims patent infringement on two similar sequences, a court must decide if the sequences have essentially the same function. This is known as the doctrine of equivalence. The requirements for protecting intellectual property rights and the sale of products with patents are not the same outside the United States and require separate applications in these countries. According to several studies, approximately 20% of all human genes have been patented, primarily by private biotechnology companies. Not surprisingly, the most heavily patented genes are typically those implicated in human health and disease. Almost 50% of known cancer genes have been patented. In some cases, genes such as BRCA1, one of the genes involved in breast cancer, have patent rights issued to multiple companies, resulting in disputes over commercial rights and ownership of such sequences. Some genes have up to 20 patents covering rights to commercial uses of the genes, as for diagnostic tests for genetic testing. See Figure 12.5 for a map of patent hotspots on chromosome 20. Figure 12.5 an illustration of patent activity on one human chromosome. Chromosome 20 is shown, with each horizontal bar representing a patent claiming a gene sequence located in that region. The labels indicate the loci of highly patented genes. You decide, would we see drug development without patents? Gene patents are here to stay, but questions remain. For instance, what happens when a single company owns a biotechnology product with potentially life-saving applications? As Table 12.4 illustrates, many companies have applied for patents on gene therapies for cancer. History shows that medical practice can be affected by patent law. For example, Myriad Genetics holds exclusive patents on BRCA1 and BRCA2, the genes that largely determine a woman's risk for breast cancer. The company demands fees from any lab that runs blood tests for the genes, a move that threatened to make the test unavailable to many women. In January 2001, the National Cancer Institute, NCI, struck a deal with Myriad Genetics that allows all NCI and NIH labs to use the test at a discount. Still, some other labs are no longer able to afford the test. A landmark decision brought by the American Civil Liberties Union against Myriad Genetics in March 2010 brought into question this ability to patent genes. The U.S. Patent Office has issued about 35,000 patents for gene sequences. It is very likely that this decision will be reversed or limited by the appeals process, but it will undoubtedly lead to a better definition of products of nature. Many in biotech see this decision as a stimulus to innovation, since manipulated by the hand of man will be better defined and lead to specifically directed products. The court held that diagnostic and screening methods that utilize gene sequences are not patentable. Will this lead to more expensive biotech products or less screening tests? Were the courts correct in their decision? You decide. 12.7 International Biotechnology Regulation. Biotechnology is a global enterprise. At the present time, the world community is still involved in preliminary negotiations about the regulation of biotechnology products. There have been some promising beginnings. The United Nations, UN, for example, has produced a voluntary code for the deliberate release of GMOs. Public comment has been solicited since it was proposed in 2000, and no final binding regulations have been agreed on. An agency of the UN, the Food and Agriculture Organization, FAO, is focusing its attention on biotechnology in developing nations, providing guidance on safety and risk assessment. In general, no consensus or clear set of commonly held standards for the import and export of biotechnology products exists. There is reason to be optimistic, however. Recent developments in Europe can serve as a model for international cooperation in the trade and development of biotechnology products. The European Union. In recent years, the European Union, EU, has begun to stand out as both a hotbed of innovation and an attractive market for biotechnology products. The formation of the EU in 1993 dramatically streamlined the process for introducing new biotechnology products to the marketplace. Today, the EU stands as a textbook example of a government body committed to promoting scientific progress. A key part of the new system is the European Agency for the Evaluation of Medicinal Products, EMEA, the equivalent of the FDA. The EMEA authorizes medicinal products for human and veterinary use. It also helps spread scientific knowledge by providing information on new products in all 11 official EU languages. Once a product has been approved by the EMEA, it can be marketed in all of the 15 countries in the EU. New drugs can now be brought to market throughout the EU in 1 year instead of 4 or 5, compared with the 8 to 11 years that the FDA usually takes to approve a new drug. Since its inception, the centralized procedure has approved more than 100 human medicines and 25 veterinary products. There is one noteworthy difference between the EMEA and the FDA. As previously discussed, the FDA is closely involved with product development from the moment a company seeks permission to conduct clinical trials. Within the EU, however, approval for clinical trials is granted by authorities in each country, not the EMEA. In many cases, the EMEA has no prior contact with the company or even knowledge of the product until it is submitted. Career Profile Quality Control Inspector. Quality control inspectors in biotechnology companies are responsible for inspecting and checking products to make sure they meet the specified levels of quality described on the package inserts, and that the product's specifications are filed with the appropriate regulatory agencies. This is a good career for a detail-oriented person who likes to work in the lab. It is an area that has a continued high rate of employment. Quality control is involved at every stage of production. Some inspectors examine raw materials to verify specified requirements, others test equipment and verify accuracy, and others test the standard operating procedures established by the company, based on FDA, USDA, EPA, and related regulations. Although a high school diploma and experience in quality control is adequate in some companies, most employers prefer an associate's degree in a technical area and a few years of experience. Quality control inspectors are often required to determine error levels in measurements, making a good math background a necessity. Good hand-eye coordination and mechanical aptitude are also required. A strong background in the regulations that govern the products of the industry is a must. Quality control inspectors with a bachelor's degree and experience can be promoted to manager. Many transfers occur within biotechnology companies from quality assurance to quality control. The Quality Assurance Department is responsible for maintaining all of the paperwork that is reviewed when any regulatory agencies show up for an inspection. A good career ladder exists in many companies from quality assurance to quality control. For review. According to the most recent polls, 2000, 75% of pharmaceutical companies in the EU are satisfied with the process as it stands. International regulations can affect U.S. companies. Some 48 million potential doses of flu vaccine were lost by the Emeryville, California company Chiron when British regulators from the Medicines and Healthcare Products Regulatory Agency, MHRA, found bacterial contamination in some lots of the vaccine. Companies that operate in the United Kingdom must follow GMP regulations that are as strict as or stricter than those in the United States. After the MHRA closed the plant in 2004, the FDA performed its own investigation and listed 20 violations in a warning letter sent to Chiron in December 2004. The company found Serratia bacteria in 9 of its flu vaccine lots. Because it could not trace where the problem started, it was forced to destroy 91 lots of vaccine. According to current federal regulations Section 211, standard operating procedures are required for the traceability of problems like these. How can other companies avoid lapses in GMP like the Chiron incident? FDA regulators recommend that firms should evaluate every aspect of production, including people, procedures, equipment, materials, record keeping, audits, and training. Current GMPs require continual updating, and companies are required to provide their employees with this training. Making a difference. Federal regulations for drug manufacture, FDA Section 211, require that all employees be trained in current good manufacturing procedures, CGMP. Biotechnology companies routinely provide training and time to make these regulations clear to their employees. The FDA monitors compliance and fines those who are out of compliance. In May 2010, the company Genzyme was fined $175 million for not fixing practices at its Alston filling plant near Boston. The FDA found tiny particles of steel, rubber, and fiber in drugs products at the plant. The company was required to shut down the plant until the problems were fixed. Compliance is part of working in the biotechnology industry and requires vigilance, but reporting problems is also a CGMP requirement. In most cases, companies are happy to change procedures that are likely to lead to the contamination of products because they want to produce sterile, life-saving products, and can't risk the disgrace of a regulatory non-compliance report. Questions and activities. Answers can be found in Appendix 1. Which U.S. agency regulates food additives? Which U.S. agency regulates GM crops? Which U.S. agency regulates water quality? Which U.S. agency regulates environmental release of genetically engineered organisms? How do patents protect drugs and devices? How have new streamlined approval processes affected the European drug market? Where are the greatest number of patent applications filed in cancer therapy? In your opinion, what should have happened according to FDA regulations to prevent the fluvirin incident? In your opinion, what is wrong with drugs being advertised for purposes other than what was specified in their FDA approval? Why are drugs often available in Europe before they become available in the United States?